There's a question that has quietly fascinated scientists, athletes, coaches, and ordinary people for decades. It lives in the space between what we observe in stadiums, weight rooms, and Olympic arenas, and what we understand in laboratories and genome databases. The question is simple on its surface, but enormously complex underneath. Are different groups of people, separated by geography and ancestry, built differently when it comes to physical strength? And if they are, how much of that comes down to the genes they were born with? This is not a question that belongs to the fringes of science. It is being actively investigated by researchers at major universities, published in peer-reviewed journals, and debated at sports medicine conferences around the world. It is also a question that gets hijacked almost immediately by sensationalism, by viral headlines promising shocking revelations about race and physical power, by content designed to provoke rather than inform. The actual science, as it almost always does, tells a story that is far more nuanced, far more interesting, and far less dramatic than any clickbait headline could ever capture. So let us start where the real story starts. Not with rankings, not with declarations about who is strongest, but with something much more fundamental, the architecture of human muscle itself. Every skeletal muscle in your body is made up of individual fibers, and those fibers come in two broad categories. There are slow twitch fibers, also called type I fibers, which are built for endurance. They are highly efficient, packed with mitochondria, resistant to fatigue, and perfectly suited for long-distance running, cycling, or sustained effort over time. Then there are fast twitch fibers, called type anthropology, which are built for explosive output. They contract faster, generate more force in a shorter period of time, and are the machinery behind sprinting, jumping, and lifting heavy weight. Most humans have a roughly equal mix of both, somewhere around 50% of each, but the baseline hides enormous individual variation. Some people are naturally closer to 60 or 70% slow twitch. Others lean heavily toward the fast twitch end of the spectrum. And that ratio, which research confirms is largely determined by genetics, has profound consequences for what kind of athlete you are likely to become. Here's where things get interesting from a population standpoint. Scientists have looked at whether these fiber type ratios differ on average across ethnic groups. The answer, based on the available evidence, is somewhat, but less dramatically than popular narratives suggest. One study comparing sedentary African and Caucasian individuals found that African subjects had approximately 8% more fast twitch fibers on average. But two other studies, using similar methods, found no significant difference at all. That is the kind of inconsistency that should make anyone pause before drawing sweeping conclusions. When three studies disagree, the honest answer is that we do not yet have a reliable, settled measurement of this difference, if it even consistently exists at a population level. What we do know is that the variation within any ethnic group is staggeringly wide. The range of slow twitch fiber percentages across all humans runs from as low as 13% to as high as 96%. That is an enormous spectrum. A black man and a white man standing next to each other in a gym might have completely opposite fiber compositions, while two brothers from the same family might be nearly identical. Genetics shapes these distributions, but it does so at the individual level far more powerfully than at the group level. This is not a rhetorical point designed to dodge the question. It is the actual scientific reality. Now, to understand why this conversation gets so complicated, you have to understand something about how genetics works in populations. Human genetic diversity is enormous, but it is not distributed evenly. Different ancestral groups carry different frequencies of specific gene variants. Some of those variants are relevant to physical performance. And the study of those variants, which ones exist, how common they are in different populations, and what they actually do to muscle, is where the real science lives. The most studied gene in this entire conversation is called ACTN3. It encodes a protein called alpha-actinin 3, which lives almost exclusively in fast twitch muscle fibers. This protein plays a direct role in how those fibers contract and how much force they can generate. The reason ACTN3 became so famous in sports genetics is that there's a specific variant, a mutation at position 577 of the gene, that essentially turns the gene off. People who inherit two copies of this deactivating variant, one from each parent, produce no alpha-actinin 3 at all. Their fast twitch fibers are missing this protein entirely. And remarkably, that is not a rare situation. About 18% of healthy white Europeans carry this deactivating variant in both copies of their ACTN3 gene. Nearly 1 in 5 people of European ancestry produces none of this protein and shows no dramatic health consequences from it. The human body has simply adapted around its absence. But what happens when you look at elite athletes? The picture changes sharply. Research published in the American Journal of Human Genetics found that among elite sprint athletes, people competing at the national and Olympic level, almost none of them carried two copies of the deactivating variant. In the female sprint group, not a single athlete who had competed at the Olympic level was deficient in alpha-actinin 3. The gene variant that makes alpha-actinin 3 non-functional was dramatically underrepresented among the fastest, most powerful athletes on Earth. This was some of the strongest early evidence that a single gene variant could influence athletic performance at the highest level. The follow-up question was obvious. If this protein matters so much for power performance, do different populations have different rates of carrying it? And the answer is yes, they do, though again, the nuances matter enormously. East Asian populations tend to have the highest rates of the deactivating variant, meaning a larger proportion of people of Asian descent produce no alpha-actinin 3. African and European populations tend to have higher frequencies of the functional variant. These are real differences in gene frequency. They are not trivial, but they describe population averages, not individual destinies. An East Asian sprinter with 2 copies of the functional variant will perform very differently from an East Asian distance runner with 2 copies of the deactivating variant, and neither of them is defined by the population average. A major 2024 systematic review and meta-analysis pulled together data from dozens of studies and confirmed that power athletes, weightlifters, sprinters, throwers, show a consistent pattern of carrying the strength-supporting variant at higher rates than both endurance athletes and non-athletic controls. This review also confirmed that the relationship is real and statistically robust. But the authors were careful to note what the data cannot tell us. Gene frequency differences between groups do not translate cleanly into performance predictions for individuals, and they certainly do not justify the kind of racial hierarchy narratives that circulate in popular media. Beyond ACTN3, there's an entire landscape of genetic variants that influence muscle strength and composition. Scientists have identified a gene called CDKN1A that appears to influence the ratio of fast twitch to slow twitch fibers. Specific variants in this gene were associated with having more slow twitch fibers across Russian, Finnish, and Japanese cohorts, 3 very different ancestral groups that were all studied together. The frequency of these variants differed between populations, which gives you a sense of how many layers of genetic variation exist across human groups, each contributing a small piece to the overall picture of physical capacity. There's also a growing body of work on what scientists call polygenic scores for muscle strength. Rather than focusing on a single gene, these scores aggregate hundreds or even thousands of tiny genetic influences into a single estimate of genetic predisposition. A major Finnish study published in 2024 using genome and health data from more than 340,000 people found that a higher polygenic score for muscle strength was associated with longer lifespan and lower risk of common diseases. This was the most comprehensive study of its kind to date, and it demonstrated something important. The genetics of strength is not a simple story about 1 or 2 genes. It is a vastly complex network of influences, most of which we are only beginning to understand. What is particularly striking about that Finnish research is what it reveals about how these genetic predispositions interact with lifestyle. The genetic advantage for muscle strength predicted better health outcomes independently of physical activity levels and socioeconomic factors. That means some people are genuinely dealt a better genetic hand when it comes to muscle health. But the research also showed that this genetic luck does not excuse anyone from the enormous influence of training, nutrition, sleep, and lifestyle. Genetics sets a ceiling, but behavior determines how close to that ceiling you get. And this principle applies equally across all ancestral groups. Now, let us turn to something more directly observable. Hand grip strength across ethnic populations. Hand grip strength is one of the most widely used measures in health research because it is easy to measure, highly reproducible, and turns out to be a surprisingly powerful predictor of overall health. It correlates with lean muscle mass, bone density, cardiovascular health, and even longevity. Scientists have measured it in populations around the world, and the results reveal fascinating variation. A large study called the Helios study measured hand grip strength in more than 12,000 participants across 6 ethnic groups living in the Netherlands: Dutch, South Asian Surinamese, African Surinamese, Ghanaian, Turkish, and Moroccan populations. The researchers found statistically significant differences in hand grip strength across these groups. But here is what the study actually showed that most people miss. The relationship between low grip strength and health risks, specifically type 2 diabetes, was essentially identical across all 6 groups. The implication is profound. Grip strength is a marker of muscle health, and muscle health predicts disease risk similarly in every ethnic group studied. The differences in average grip strength between groups did not change the underlying biology of why strong muscles protect against disease. A 2025 population-based study in Nigeria explicitly called for population-specific normative values for hand grip strength, acknowledging that racial, genetic, and geographic differences are real and need to be accounted for in clinical practice. This is the practical consequence of these differences. Not a ranking of who is physically superior, but a recognition that global medical standards developed primarily on white European populations may not apply accurately to every group. That is a clinical equity issue, not a hierarchy. A multi-ethnic study comparing Indians, Malaysians, Chinese, Dutch, Africans, Iranians, and Polish individuals on grip strength found differences that tracked partly with hand size and body proportions, variables that are themselves influenced by both genetics and environment. Taller people with larger hands generally have stronger grips, and average body dimensions differ across populations for a complex mix of genetic, nutritional, and historical reasons. Disentangling the genetic component from the environmental and developmental component is one of the hardest problems in this entire field. This brings us to one of the most important and underappreciated aspects of this entire conversation: the difference between genetics and race as a category. When scientists study gene frequencies in populations, they are working with something real and measurable. Specific DNA sequences either exist in a genome or they do not. Their frequencies in a given ancestral group can be counted. This is rigorous science. But the racial categories that get used in popular discussions, black, white, Asian, and so on, are social and political constructs that do not map cleanly onto the population groupings that genetics actually describes. Consider what the word Asian encompasses. It includes populations as genetically distinct from each other as Japanese, Pakistani, Thai, and Kazakh people. The genetic distance between a person from Japan and a person from India is comparable to the genetic distance between a person from Ireland and a person from Saudi Arabia. Using Asian as if it were a coherent genetic category for studying strength genetics is scientifically meaningless. The same problem applies to black as a category that groups together West Africans, East Africans, and African Americans, who have extremely different ancestral histories and genetic profiles. And white covers everyone from Finns to Spaniards to Russians, populations that differ substantially in allele frequencies for many genes. This is not a political point. It is a scientific one. The most meaningful genetic differences relevant to muscle performance exist at the level of specific ancestral populations or individual variants, not at the level of the broad racial categories that dominate popular discourse. A West African population from coastal Ghana and an Nilotic population from South Sudan differ enormously in muscle fiber genetics, average body proportions, and metabolic profiles. Lumping them together and calling them both black for the purposes of strength genetics, research is not just imprecise, it actively obscures the real patterns. And yet the research does find some consistent signals when populations are compared at a broad level. The overrepresentation of West African ancestral athletes in sprinting and jumping events has been documented extensively. The dominance of East African athletes, particularly from Kenya and Ethiopia, in long-distance running is one of the most statistically remarkable patterns in all of sports. Researchers have proposed multiple explanations for this, ranging from muscle fiber type ratios to tendon length to limb proportions, to altitude adaptations, to cultural emphasis on running from childhood. The honest answer is that it is probably some combination of all of these factors, and the exact weighting of each contribution remains actively debated. What is notable is that even in the most intensively studied case, the dominance of Kenyan long-distance runners, genetics does not tell the whole story. Studies of Kenyan athletes have found that while certain gene variants relevant to endurance are somewhat more common in the Kalenjin ethnic group, which produces a disproportionate number of elite runners, the frequencies are not dramatically different from those found in other populations. What is different is the training environment, the altitude at which Kenyan children grow up, which stimulates blood oxygen carrying capacity, the cultural valorization of running, and the economic incentive that a running career provides. Strip away those environmental factors, and the genetic signal alone would not predict Kenya's dominance in global distance running. This is a pattern that repeats throughout the science. Every time researchers dig into a seemingly clear genetic advantage for a particular population, they find the environment intertwined with it at every level. Muscles are not simply executing a genetic program. They are responding to training, to nutrition, to hormonal signals, to the cumulative effects of a lifetime of physical experience. Two people with identical ACTN3 genotypes, but radically different training histories will perform very differently. Two people with different genotypes, but similar elite training programs may end up remarkably close in performance. The gap between genetic potential and realized performance is enormous, and it is filled almost entirely by environmental factors. A 2025 review published in the Journal of Integrative Social Sciences made this point explicitly. It cautioned that the observed disparities in muscle mass and strength between ethnic groups need to be understood through the lens of biological, genetic, physiological, and evolutionary factors, all working together, not through any single explanatory lens. It also noted something clinically important. Current medical standards for assessing muscle health were largely developed on white European populations, and applying them universally may lead to miscalculating health risks in populations like Black and Asian individuals. Getting this science right is not just an academic exercise. It affects how doctors assess sarcopenia, the age-related loss of muscle, how they evaluate frailty in elderly patients, and how they interpret strength measurements in people who differ from the populations on which those standards were built. The question of heritability is worth addressing directly because it often gets misunderstood in popular discussions. Heritability estimates for muscle strength typically range from around 50 to 80% in twin studies. That sounds like a lot, and it is. But heritability does not mean what most people think it means. It does not tell you that 50 to 80% of your strength is determined by your genes in any absolute sense. It tells you that, in the specific population studied, under the specific environmental conditions of that study, 50 to 80% of the variation in strength between individuals is associated with genetic variation rather than environmental variation. Change the environment dramatically, for instance, put everyone through the same elite training program or give everyone access to the same nutrition, and the heritability estimate can shift substantially. This distinction matters enormously for how we interpret cross-population comparisons. When researchers compare average strength between ethnic groups, they are comparing people who live in very different environments, eat very different diets, have very different histories of physical labor, access very different levels of healthcare, and face very different levels of chronic stress. All of these factors influence muscle strength independently of genetics. Teasing apart the genetic contribution from these environmental differences is extraordinarily difficult, and most studies that attempt it acknowledge the limitations of their methods. There's also the question of muscle quality versus muscle quantity. A 2025 review noted that black populations, on average, tend to have higher baseline muscle mass than white populations, but that this does not straightforwardly translate into proportionally greater strength. The relationship between muscle size and force production depends on factors like fiber type composition, neuromuscular efficiency, meaning how effectively the nervous system recruits and coordinates muscle fibers, tendon mechanics and muscle architecture, including the angle at which fibers are attached to tendons. Two muscles of identical size can produce very different amounts of force depending on these architectural details. This complexity means that simple measurements of muscle mass or even grip strength do not fully capture the multidimensional reality of physical strength. One area where population-level differences in genetics may have clearer implications is in medicine rather than sports. Differences in muscle fiber composition and muscle quality between ethnic groups are relevant to understanding why certain diseases present differently across populations, why sarcopenia progresses at different rates, and why the same drug or intervention may have different effects in patients of different ancestral backgrounds. The scientific interest in ethnic differences in muscle genetics is, at its core, most clinically valuable, not for deciding who wins a weightlifting competition, but for understanding how to keep people of all backgrounds healthier as they age. The story that genuine science tells about strength genetics across human populations is one of variation, enormous, continuous, multidimensional variation. Some of that variation has a genetic basis. Some of it reflects differences in population history, natural selection pressures, and the frequency of specific gene variants across ancestral groups. Some of the most studied variants, like ACTN3's R577X polymorphism, show clear and replicable differences in frequency between populations and clear associations with athletic performance at the elite level. But the effect sizes, the actual magnitude of advantage conferred, are modest when measured against the full spectrum of human variation. No population has a monopoly on strength-relevant genetics. No ancestral group is categorically weaker or stronger than another. What populations do have is different distributions of many, many small genetic influences, each one contributing a tiny piece to the overall puzzle of physical capacity. Layered on top of this genetic variation is an equally vast layer of environmental influence: diet, training, culture, altitude, history of labor, access to resources, chronic stress, and countless other factors. The intersection of these two layers, the genetic and the environmental, produces the actual athletes we see competing in the real world. The actual workers lifting and carrying, the actual patients whose muscle health determines how well they age. When scientists compare populations, they are sampling from these complex, overlapping distributions. They find real differences in averages. They document genuine variation in gene frequencies. They confirm that some genetic variants relevant to muscle performance are more or less common in certain ancestral groups. None of this is hidden or controversial within the scientific community. What is controversial, and what the evidence does not support, is the leap from these modest population-level frequency differences to any claim about the inherent superiority or inferiority of an entire group of people. The Human Genome Project famously established that all humans share about 99.9% of their DNA. The remaining fraction of a percent accounts for every observable difference between any two people on Earth: eye color, height, disease susceptibility, and yes, certain aspects of muscle physiology. Within that tiny fraction of difference, the amount attributable to broad racial categories is smaller still. More genetic variation exists within any so-called racial group than between groups. This is not a political talking point. It is one of the most replicated findings in all of population genetics. The science of strength genetics is genuinely exciting. It is revealing how specific molecular mechanisms, proteins in muscle fibers, regulatory genes controlling fiber-type ratios, polygenic networks influencing muscle quality, vary across individuals and populations in ways that have real consequences for health and physical performance. As sequencing technology becomes cheaper and cohort studies grow larger, researchers are beginning to map this variation in increasing detail. Within the next decade, polygenic scores for athletic potential and muscle health may become genuinely useful clinical tools, helping doctors identify individuals at high risk for muscle-related diseases years before symptoms appear. But the translation of this science into public discourse is where the danger lies. Every genuine scientific finding about population differences in strength-relevant genetics will be extracted from its context, stripped of its caveats, and used to construct narratives that the science itself does not support. This has happened before, and it will happen again. The appropriate response is not to pretend the variation does not exist. That is scientifically dishonest. The appropriate response is to understand it accurately, as population-level frequency differences in a complex genetic landscape, shaped by deep evolutionary history, operating within bodies that are simultaneously being shaped by diet, training, culture, and environment, producing individuals whose physical capacities cannot be meaningfully predicted from their ancestry alone. Every elite powerlifter, sprinter, gymnast, or distance runner on the planet is a specific individual with a specific genetic makeup, a specific training history, a specific nutritional background, and a specific set of choices made over years of effort. No gene test or ancestral category tells you what any individual is capable of. The ACTN3 genotype of every Olympic sprinter is known and has been published, and yet the gold medal still goes to whoever crosses the finish line first, not to whoever had the most favorable genotype profile. The gap, when it comes to strength genetics and human populations, is not between races. It is between the complexity of the actual science and the simplicity of the stories we tell about it. Closing that gap, understanding the real biology in all its nuance, is not just intellectually satisfying. It is essential for medicine, for sports science, for clinical practice, and for the basic human project of understanding ourselves clearly and honestly. That is where the science stands today. Not with a ranking, not with a definitive hierarchy, but with a landscape of variation that is real, measurable, and endlessly more interesting than any viral headline could capture.